You haven't opened the web,
no,
right? Webinar, not yet, right?
Yeah, I'll do it right
now.
Okay.
Okay.
Okay, good afternoon, everyone. Welcome to the 34th MSPP Scientific Meeting Symposium for Cancer Research. I'm Chichita, a senior lecturer from Jeffrey Cha School of Medicine and Sciences, a member of a scientific committee. And today we have our honorable speaker, Dr. Alan Yulweli, who is our colleague from School of Pharmacy, Monash University, Malaysia. Please let me introduce a brief introduction about Dr. Alan. Dr. Alan Lee obtained his PhD from University of Hong Kong. He is currently a senior lecturer at the School of Pharmacy, Monash University, Malaysia. Current focus, his research is to understand the function of axon guidance molecules, Semaphorins, and plexins in the cell migration and invasion. Axon navigation, cellular differentiation, neuronal degeneration, and brain tumor development. His research team is recently revealed the tumor suppressor functions of this semaphorin 5A in the human glioblastomas and is currently exploring the therapeutic potential of it. Dr. Lee has published his research in the leading journals in neuroscience, cancer biology, and he served as an editorial board member and reviewer of the many reputable journals. Today, we will get an opportunity to hear from Dr. Alan about his research entitled Exploration of the Tumor Suppressor Function of Semaphorin 5A in Brain Cancer Treatment. I'm sure we all have benefits from his talk, and if you have any questions, please indicate in the Q&A box, and Dr. Alan will answer after the talk. Without further ado, I would like to invite and welcome Dr. Alan for his presentation. Dr. Alan, I will over to you.
Thank you very much, Dr. Ta, for your kind introduction. So I also take this opportunity to thank the organizing committee for giving me this opportunity to give a talk in this symposium. Share my slides. Okay, so what I'm going to do this afternoon is to share with you some of my findings regarding the tumor suppressor functions of the molecule semaphorin 5A and how is it involved in as a potential therapeutics for treating brain cancers. So in this case, for brain cancers, I'm referring to mainly gliomas. Okay, so for gliomas, they're actually the most common types of CNS tumors. Okay. And there are actually different types of gliomas, and among them, for astrocytomas, they actually account for 60% of all primary brain tumors. Okay, and according to the WHO classification, they can be classified into 4 different grades, from grade 1, which is low grade, to grade 4, which is high grade. And grade 4 is also referred to as glioblastoma multiforme, okay, or what we call the GBM in short. Okay. So GBM for the typical treatment is actually involved surgical resection of the tumor if the tumor is reachable, okay, and then followed by standard radio and chemotherapy. Okay, but it seems that this traditional kind of treatment is actually ineffective, okay, mainly because of the fact that these glioblastoma cells, they are actually highly invasive, okay, and they are resistant to chemo and radiotherapy. So that's why even before surgical resection of the tumor, some of these glioblastoma cells, they have already invaded and infiltrate to the nearby region. Okay, and because of the fact that they are actually resistant to chemo and radiotherapy, so that's why they tend to proliferate, okay, even after the tumor has been removed. So that's why they will lead to a high recurrence rate even after radical surgical resection of the tumor. And it turns out that the patient prognosis becomes very poor. Okay, and the median survival of glioblastoma patients range from 12 to 18 months after initial diagnosis. Okay. And if you take a look at the 5-year survival curve, okay, if you take a look at the GBM, that means the grade 4, the most malignant form of glioblastoma, the 5-year survival is actually as low as 5%. Okay. And even if you take a look at the average of grade 2 to grade 4, the 5-year survival actually hover around 30% for the past few decades. So apparently there is an urgent need for us to look for alternative form of treatments, okay, in order to treat this debilitating disease. So now, if we take a look at the infiltration pattern of these glioblastoma cells, as I mentioned earlier, they are actually highly invasive. And it seems that they show a preference to invade through major axonal fiber tracts, for instance, the corpus callosum. Okay, and that would actually end up with what we call a glioma butterfly, okay, as a clinical manifestation. Okay, so in fact, back in the neuronal development, these axonal fiber tracts, okay, they also travel from one hemisphere to the opposite hemisphere, okay, in order to connect neurons from the both hemispheres in the brain. And it turns out that this process is actually tightly regulated by whole families of axon guidance molecules. So this raised the possibility that these axon guidance molecules may somehow be also involved in the regulation in the infiltration of glioma cells. And today, there are a total of 4 different families of these guidance molecules that have been reported, namely Semaphorins, LIT, leptin, and ephrin. And they work together with their receptors, okay, to serve as either attractive or repulsive cues, okay, to guide the movement of these neurons. Okay, so, and chances are that this particular process may also be involved in the guidance of cancer cells, okay, in their infiltrative behavior. Okay. And among these axon guidance molecules, semaphorin and plexins actually constitute the biggest family. If you take a look at this slide, so today a total of 8 different families of semaphorins have been reported, and all of them share a common sema domain at the N terminus. Okay, and it turns out that these semaphorins, they actually show a promiscuous interaction with their receptors plexins. And today, a total of 4 families of plexin have been reported, namely plexin A, B, C, and D. And for our lab, we are particularly interested in class 5 semaphorin, particularly semaphorin 5A, mainly because of the presence of this thrombus bonding domain, okay, in the extracellular moiety. And it turns out that these TSP domain, they are closely related to the regulation of cancer progression. And for the receptors for SEMA5A, they're actually the plexin B3. Right? And in addition to having plexin as the receptor, semaphorin would also engage some other molecules as co-receptors. For instance, neuropilins, ERB2, CMAT, okay, and even EGF receptors, PDGF receptors. So as you can tell, some of these receptors are actually receptor tyrosine kinases. They're closely related to the regulation of cancer development. Okay, and it is actually through this complex kind of interaction between semaphorin and their co-receptors that would lead to the trigger of a whole array of signaling pathway, leading to the regulation of not only axon guidance process, but also a whole array of physiological processes, including cell migration, angiogenesis, and even cancer progression as well. So as I said just now, our lab is interested in semaphorin 5A and plexin B3, this ligand receptor pair. Okay? And in fact, when we first isolate plexin B3, we actually perform an expression profiling experiment. And we found that both SemaPhin 5A and plexin B3, they are expressed in a whole range of glioblastoma cell lines, okay, at both the transcript level as well as the protein level. And strikingly, when we try to measure the level of SemaPhin 5A protein, okay, in clinical samples of glioblastoma, okay, that we obtained from our collaborator from hospital, we found that there's a gradual reduction in the level of expression of SemaPhin 5A protein from low grade all the way to high grade. Okay, if you take a look at the expression level of SemaPhin 5A protein in grade 4, the GBM, there's actually a significant reduction, up to 80% reduction of the expression level of SemaPhin 5A protein. So apparently this loss of SemaPhin 5A protein expression, particularly in high-grade glioblastoma, actually correlate with the increasing infiltration as well as the tumorigenic properties of these glioblastoma cells. Okay, so this actually raised the questions whether SemaPhin 5A could actually serve as a tumor suppressor gene. Okay, so that's why with this loss of the expression, it may actually lead to the increasing tumorigenic properties of these cancers. Okay, so in order to test this hypothesis, we actually make use of the gain of function as well as a loss of function approach. So with the gain of function, we try to force express SemaPhin 5A in glioblastoma cell line. As you can see here, we focus on the field bar. So upon overexpression of SemaPhin 5A, there's actually a corresponding reduction or inhibition of cell proliferation. Okay. And by contrast, if we silence the endogenous expression of SemaPhin 5A in glioblastoma cells using the siRNA approach, we found that the downregulation of endogenous SemaPhin 5A actually leads to a significant increase in the invasion, okay, in the invasive properties of these glioma cells. Okay, as you can see here, there's a significant increase in cell invasiveness. So based on these findings, we can conclude that SemaPhin 5A most likely serve as a tumor suppressor protein. Okay. And this leads to our working hypothesis that if SemaPhin 5A really serves as a tumor suppressor protein, okay, with its downregulation, particularly in high-grade glioblastoma, that would actually lead to a loss of its tumor suppressor function, and that would lead to the enhancement of brain cancer development and progression. So based on this working hypothesis, of course, there's still some unanswered questions that we need to tackle. The first one is, what is the mechanism of the tumor suppressor function of SemaPhin 5A in glioblastomas? And the second question is, what is the mechanism of SemaPhin 5A downregulation that you observe in glioblastoma, okay, particularly the high grade? So for the first one, okay, in terms of the mechanism of the tumor suppressor function of SemaPhin 5A in glioblastoma, we look at various hallmarks of SemaPhin 5A in glioblastoma properties, namely migration, invasion, proliferation, tumorigenicity, as well as differentiation. And the approach that we use is actually to try to overexpress, okay, SemaPhin 5A by making use of the genetic approach, or alternatively, we can always exogenously supply SemaPhin 5A protein as a ligand to stimulate the plexin B3 receptor. So in terms of the evaluation of the effect of SemaPhin 5A on glioma cell migration, we actually make use of the scratch wound healing assay. So you can see here, we make a scratch on a conference layer of glioblastoma cell line. You can see that for cells that are untreated, okay, they actually manage to migrate and repair this gap, okay, within a matter of 24 hours. Whereas for cells that have been treated with SemaPhin 5A, as you can see here, there's a significant inhibition of the migration of these cancer cells. And for all those glioblastoma cell lines that we have characterized, okay, they can actually cause SemaPhi can actually cause an inhibition of down to 50% of the migration properties of these cancer cells. And similarly, when we measure the effect of SemaPhi on glioma cell invasion by making use of a transfer assay, okay, this is a message-derived transfer assay. Again, we found that upon SemaPhi reconstitution of expression, okay, that actually lead to a 40% reduction, okay, in the invasive properties of these glioblastoma cells. So in terms of the mechanism behind this inhibitory effect of SemaPhi on glioma cell motility, we actually found that upon SemaPhi reconstitution, there's actually a significant reduction in the level of active form of RAD1, that means the RAD1 GTP. And it turns out that RAD1 GTP actually play a very important role in terms of the regulation of cell migration, okay, in the sense that RAD1 actually promote the remodeling of the actin cytoskeleton, leading to the extension of this cellular process. Okay, this is actually the very first step of cell migration. And at the same time, we also found that SemaPhi stimulation would actually disrupt the Winkelin positive focal adhesion as shown here. Okay, so this is before treatment. Okay, and as early as 15 minutes after SemaPhi stimulation, we found that all these focal adhesion are actually lost. Okay, and it turns out that Winkelin positive focal adhesion, they actually play a very important role in the integrin-mediated cell adhesion. And that actually, again, play a very important role in terms of securing the cell with a foothold onto the substratum, so that, at the new position after the extension of the cellular process, okay, and before the cell can actually pull itself forward, okay, to move on to the next location. Okay, so that's why for these two mechanisms, they are likely to contribute to the inhibitory effect of SemaPhi in terms of glioma cell motility. And in terms of the effect of SemaPhi on glioma cell proliferation, we make use of the MTS cell viability assay. As you can see here, over a course of 6 days, for the control cells, they actually show a progressive increase in the cell number. Whereas for cells treated with SemaPhi, as you can see here, there's a 55% reduction in the number of viable cells. Okay? And when we perform the cell cycle analysis, we actually found that there are significantly fewer number of cells that manage to make their way to the S phase, suggesting that SemaPhi actually will induce a G1S cell cycle arrest. Okay, that means the cells cannot progress from G1 to S phase. And in fact, when we perform the biochemical analysis, we actually found that there's a corresponding reduction in the level of the cell cycle related protein, CDK4, as well as cyclin D1, that play an important role in the G1S phase transition. And next, we look at the effect of SemaPhi on tumorigenicity of glioma cells by making use of the soft agar colony formation assay. And as you can see here, SemaPhi again would actually significantly inhibit anchorage independent growth, okay, by 55% as compared to the control. And last but not least, we also look at the effect of SemaPhi on the differentiation stage of these glioblastoma cells. So if you focus on the top panel over here, so for glioblastoma cells, they normally adopt what we call the anaplastic morphology. So that means they actually appear as an undifferentiated cell. Okay? But upon SemaPhi stimulation, we found that there's a remodeling of the actin cytoskeleton, leading to cell collapse, okay, and also with concomitant induction of process outgrowth as shown here. Okay? So if you take a look at the morphology, they actually resemble astrocyte. Okay? But it turns out that SemaPhi not only induce morphological differentiation, okay, along the astrocytic lineage, but in fact, when you try to measure the level of the astrocytic marker GFAP, okay, as shown in this immunofluorescence staining, you can see that before SemaPhi stimulation, basically you don't see much GFAP staining. But upon SemaPhi stimulation, you can see re-expression of GFAP, which is the astrocytic marker. Okay, and it is also confirmed by using Western blot analysis as well. Okay, so suggesting that SemaPhi actually induced astrocytic differentiation. Okay, this actually fit into the hypothesis that glioblastoma cells, they actually have their origin from astrocyte. Okay, it's just that by undergoing what we call the differentiation process, these astrocytes gradually lose these differentiation properties and become anaplastic in morphology. Okay, and now our findings suggest that SemaPhi actually reverse this particular process, so that the glioblastoma cells now start to reacquire the astrocytic properties, okay, and also become less tumorigenic. Okay, so the next big question that we need to answer is, what is the mechanism that will contribute to the loss of SemaPhi expression, particularly in high-grade glioblastoma? Okay. So of course, the most straightforward approach would actually to try to measure the transcript level of SemaPhi in these brain cancer cells. But what we found was that there's no significant change in the message level of SemaPhi, both in the cell line as well as in the clinical samples from patients. Okay. And next, we try to look for any mutation in the SemaPhi gene that may lead to its dysregulation. But unfortunately, we didn't find any mutation. Okay, so based on this observation, we are actually left with a choice. Okay, basically, the downregulation of SemaPhi most likely will be due to post-transcriptional dysregulation. So in fact, when we perform the sequence analysis of SemaPhi gene, we actually found that there's a significant increase in proportion of the SemaPhi transcript that show alternative splicing. Okay, so as you can see here, I would like to draw your attention to these two splice variants, namely SemaPhi DEX4 and DEX11. Particularly for DEX4, as you can see here, exon 4 is actually alternatively spliced out from the transcript. Okay. And it turns out that by excluding exon 4, that will actually lead to a shift in the translational reading frame, leading to the formation of premature stop codon, and that would actually translate into a prematurely truncated protein. Because for wild-type SemaPhi, it consists of 23 exons. Okay, if they are correctly spliced, it will lead to the formation of a protein that is of about 120 kilodalton. But for this splice variant, in which exon 4 is excluded, the protein becomes truncated, and the size is only 11 kilodalton. Okay, and we try to analyze the percentage of these aberrantly spliced variant, okay, that would lead to the formation of truncated protein. We found that they actually constitute to up to 60%, okay, among all the SemaPhi transcript. But of course, we would still want to know that, say, whether this truncated SemaPhi protein will still be able to preserve its tumor suppressor function. So that's why we try to clone this splice variant and try to express them as protein. But to our surprise, we actually failed to detect these truncation variants. Okay, as you can see from the Western blot analysis over here, we can hardly detect the presence of this splice variant, okay, in which exon 4 was deleted. Okay, so but the thing is, when we try to incubate the cells with a proteasomal inhibitor, which is MG132 in this particular case, we found that upon treatment for 12 hours, we start to see the presence of this truncated version of SemaPhi. Okay, and when we try to perform immunofluorescence staining, okay, again for the MG132 treated cells, okay, for SemaPhi, we also found the presence of this truncation mutant, okay, at around 12 hours, okay, and also when we also try to co-localize with the proteasomal marker, which is a 20S proteasomal marker, we actually find co-localization between this SemaPhi mutant and the proteasomal marker, suggesting that this truncation mutant, okay, apparently is unstable. This particular protein is unstable, and it is targeted for proteasomal degradation. So that's why we actually hardly detect the presence of this particular mutant. Right. So that's why this, as I mentioned just now, because these splice variant that would produce truncation SemaPhi, they account for up to 60% of all the SemaPhi transcript. So that's why they likely would contribute to the downregulation of SemaPhi protein that we observe in high-grade glioblastoma. But of course, it's not the end of the story. As I mentioned earlier, we actually observe up to 80% downregulation of protein, okay, in the clinical samples of glioblastoma. So there must be some other alternative mechanism that contribute to this. So when we analyze the 3' untranslated region of the SemaPhi gene, we actually managed to identify 3 putative binding sites for the microRNA 21. Okay, and as you know, some of you may know that MIR21, okay, is actually highly upregulated in glioblastoma cells, okay, and also in many other cancer types as well. So that's why we suspect that MIR21 may actually bind to the 3' untranslated region of SemaPhi and cause a downregulation of protein by translational repression. So to test this hypothesis, we actually tried to overexpress MIR21 in glioblastoma cells. And what we found was that as early as 24 hours after upregulation of MIR21, there's a significant downregulation of endogenous SemaPhi in these glioma cells. And by contrast, if we put in the inhibitor for this MIR21, we actually observe an increase in the expression level of the endogenous SemaPhi. So taken together, these findings strongly suggest that SemaPhi is actually subjected to MIR21 regulation, and that would actually cause the downregulation of protein without changes in the messenger RNA level. So that's why, in summary, what we found in this piece of study is that SemaPhi protein is actually downregulated by 80% in high-grade glioblastoma, okay, from our clinical analysis of the specimen. And we constitute the SemaPhi expression in these cells can actually restore its tumor suppressor function. And in terms of the mechanism that causes the downregulation of SemaPhi is we actually observe 60% of SemaPhi transcripts that are aberrantly spliced, okay, that will actually result in the formation of truncated proteins. Okay, and the SemaPhi expression is also downregulated by MIR21 as well. So that's why, in summary, SemaPhi is confirmed to be a tumor suppressor, and it can be developed as a novel diagnostic marker and therapeutic targets in glioblastomas. In terms of future direction, we have already mapped the domain on SemaPhi that contribute to this tumor suppressor function, and we are in the process of developing it as an experimental therapeutics by encapsulating the protein in a biopolymer for controlled drug release. And we also are interested in exploring the function of other Semaphorins and plexins in brain tumor stem cells that potentially can initiate gliomas. Okay, so this is the acknowledgement slide, okay, for all the hard work done by the lab members and also the financial support by the funding agency. Okay, thank you very much. Happy to take questions if time permits.
Thank you very much.
Thank you.
Thank you very much, Dr. Allen, for the very informative and comprehensive talk. We have a few questions, and first question is, what is the reason for preferentially invasion into the corpus callosum?
Okay, so that's a very good question. So in fact, this is a clinical observation that these glioblastoma cells, they always tend to invade along fiber tracts. Okay, so but if you want to know exactly why, I can't really give you an answer because it's just an observation. But the possibility is that these glioblastoma cells, they always need to have something to adhere to before they manage to migrate to a long distance. For short distance, I think it shouldn't be a big problem. But for long distance infiltration, they do need to cling on to something. Okay, so that's it's actually the whole reason why we started this project, because of the observation that these glioblastoma cells, they actually tend to attach to the fiber tracts. Okay, and that actually leads to the whole stories of the involvement of Axon guidance molecules in the regulation of glioblastoma cell properties of high infiltration. Right?
Thank you. And next question is, how is the gene mutation study done?
Okay, so gene mutation study, basically, we actually took a different approach because conventionally, I think scientists or researchers, they would actually sequence the genomic sequence. Okay, but for our case, we actually took a shortcut. Okay, instead of sequencing the genomic DNA, we actually sequenced the cDNA. Okay, because even before these genetic studies, we actually have already performed in silico analysis, and based on the information from the database, it seems that there's no sign that there are mutations, okay, in the SemaPhi gene. So that's why we actually took a shortcut by sequencing the cDNA rather than the genomic DNA, and that actually led to our discovery of all these alternatively spliced variants, and that actually contribute to the formation of these truncated proteins that are actually unstable and being targeted for degradation.
Thank you very much, Dr. Allen, and all the participants Dr. Allen for highlighting the therapeutic potential and role of the Axon guidance molecules and its further therapeutic potential for this. So without further ado, we have to move to another section. So I thank you all the presenters and as well as all the participants for this section. So we will continue with the section, oral presentation. Thank you.
Thank you very much, Dr. Chichita, for handling the earlier session. We will straight away move on to oral presentation session this afternoon. Before we begin, I'd like to ask all the oral presenters to raise your hands so that we could add you into the panel room so that you'll be able to take the questions by the judges.